Dr. Donate WEGHORN, thanks so much for coming on to evolutionsoup from your home in Barcelona, Spain. You are group leader at the Center for Genomic Regulation, Barcelona, Spain, studying mutational processes in human populations. In November 2025, you published research on a mutational hotspot that your team identified within the human genome, an explosively dangerous area affecting brain function as well as the onset of cancer. Welcome to the show, Donate. Well, the paper has been out for a few months now, so how does it feel to finally be able to get this work published? And what have the initial reactions been like?
Yes, so thank you very much for having me on the show. I'm really delighted to be here. And yeah, so the paper, well, it was a lot of work. So I'm very happy to finally see it out. We've worked on it for, yeah, almost 5 years with different people working on it, contributing, also a collaborator from Boston. And it was very nice teamwork because we kept adding more results and findings. And the reactions were actually quite unexpected. So it seems that the public has taken quite some interest in the findings. Although I feel like the main interest is not so much in the mutation process that we found, but in like what does it do, which genes are most affected, which I understand because this is very easily relatable, right? Because we're all humans and we're very interested in diseases.
Before we hear about this incredible work identifying these mutation flashpoints, let's just hear a little bit about your background. Donate, where did you grow up, and what were the influences and fascinations that led you to where you are now?
Yeah, so I grew up nearby Bonn in Germany, which, so Bonn is a city of around 350,000 people, and 10% of those are the student body. So it has a very old and quite large university, which is where I studied physics. And yeah, Bonn is actually also the former capital of Germany was the capital before the fall of communism. And so during my studies, I had a focus on cosmology because that was the initial driver, astronomy that motivated me to study physics. But then during my master's thesis or diploma thesis, as it was called still at the time, I realized that this was not what I wanted to be working on for the rest of my life. So then I switched to the Department of Theoretical Physics at the University of Cologne to start working on population genetics or evolutionary phenomena. Because there's a very easy kind of natural segue from statistical physics into population genetics because it mathematically is the same or much of it is the same framework. And biology had always been a very strong interest of mine in high school and even before, more so even than physics in some way. And then after my PhD, I went on to Boston to do my postdoc there at the Harvard Medical School. I joined a lab that did a lot of statistical genetics, so working on disease associations, but they also were pretty strong in terms of mutational processes and modeling evolution. And then I carved out my niche there, working on cancer evolution and cancer genomics, because that's why I went there. That's what I wanted to work on in that lab. And then finally, in 2018, I came to Barcelona directly from Boston to start my own group here at the Center for Genomic Regulation, or CRG, where I lead a team of, at the moment, 6 people.
Donata, before we explore this important new research, can you give us a snapshot of your work at the WEGHORN lab, as well as an overview of DNA research in general and why monitoring mutations is so important?
Yes, with pleasure. So we're a pure dry lab in the CRG, which is a bit rare there because out of the 32 groups, there are only 3 that are purely dry. So we're surrounded by lots of experimentalists. But we do focus our work on modeling and computational biology. And all of the work in the lab revolves around evolution. we apply evolutionary theory to two different biological kind of settings or systems. One is the evolution of species, where we usually take quite an anthropocentric view, but we also look at the grand phylogeny. And then on the other hand, we do a lot of work on cancer evolution. And maybe this is not obvious to everyone, but basically the entire theoretical machinery that one uses to describe evolution, which is that of population genetics, can be applied in these two contexts. So it's practically the same. So in the context of humans or species evolution, it's probably quite obvious that a population of individuals, the individuals in that population would be the entire organisms, right? So the people, if it's humans. In the context of cancer, we're looking at the evolution of what starts out as a single cell and then acquires so-called driver mutations that then enable the cell to start growing uncontrollably into a large tumor mass consisting of millions of cells. And then we use the same framework, but the individuals now in the population are the cells themselves, right? And that's basically why we can work in these two realms and move over quite easily. And then, so when you want to study evolution, the first thing that you need to understand is always mutations, because mutations generate the substrate of evolution. They generate the genetic heterogeneity in the population. And without mutations, therefore, there would be no evolution. And so then once you have a good grasp of what determines the probability of mutation at any given locus in the genome, for example, you can start to ask questions about whether that locus might be evolving under selection, right? And by selection here, I mean just Darwinian selection, so the survival of the fittest, which is probably a concept that's very familiar to your regular viewers. And so what selection does, it differentiates between more and less well-adapted individuals in the population, or more or less fit individuals, depending on or based on the genetic differences between the individuals. And these are clearly generated by mutations, and depending on where mutation lands, it can produce a big difference in fitness between individuals, or it can be completely inconsequential. And if we think about kind of what's more likely, it's constructive to consider the fact that we have about 1.2% of our genome that consists of so-called protein coding sequences. And these are really the most important parts of our genome. And if a mutation lands there, there's a very high chance that it would cause a problem or a disease. And so again, if we want to model selection, we need to understand mutation. But this is where all the problems start, the first set of problems one has, because the mutation rate is extremely heterogeneous across the genome, and it depends on a lot of different factors. And because of that, much of the work that we do in the lab really goes first into building proper probabilistic models of mutation rate. And I can give you an example. So if you imagine a DNA segment that consists like of 3 bases, let's say adenine, cytosine, and guanine, right, which is 3 of the 4 bases, and that we will call a trinucleotide. And now the probability that the central cytosine in that trinucleotide will mutate depends actually very strongly on the 2 neighboring bases that it is embedded in. And if I were to just exchange, let's say, the G by a T, make it a thymine, I could already change the probability of the cytosine to mutate by up to more than 10-fold. So it's really very, very different. What is the probability to mutate at any given position on the DNA sequence? And besides this kind of sequence dependence, there's also a more kind of general modulation of the genome-wide mutation rates that depends on a lot of epigenetic factors. And a proper mutational model tries to account for all of this mutation rate variation together, right? So to be predictive. And the project we're going to talk about today basically grew out of such an endeavor to try and understand the mutation rate and its variation across the genome better.
In November 2025, you and your team published a paper describing your discovery of a vulnerable section of human DNA that you dubbed a mutational hotspot with very important biomedical implications. So, how did you and your team detect these hotspots?
Yeah, so indeed, we took a deeper look at the mutation rate in certain regions of the genome, and in this case, the transcription start site. So the transcription start site, first of all, maybe I should explain that a little bit. We call it the TSS. It marks the beginning of so-called protein coding genes. And upstream of the TSS, you would have the so-called promoter region, which is a region in which many transcription factors assemble when the downstream gene should be transcribed. And then when the required kind of ensemble of transcription factors is bound at the promoter of a gene, they can recruit a bigger enzyme, which is called the RNA polymerase II, because it polymerizes or synthesizes RNA. In this case, the messenger RNA that is produced from the protein coding gene. So the RNA polymerase II starts at the TSS and wanders downstream into the gene body in order to generate this messenger RNA molecule, from which then in the following steps, the protein is being translated. For many genes, the downstream region starts with a so-called untranslated region, or UTR, which doesn't contain any encodings for amino acids. And then following that, there is the first coding exon, which contains the amino acid sequence of the start of the protein, right? And then there could be introns and further exons coming further downstream. So basically, these are the two sides of the TSS. And we found that on both sides, upstream in the promoter region, as well as downstream in the UTR and the coding exon, there's a huge excess in the human germline of mutations compared to the regional average mutation rate. And that was kind of very surprising because before that had been only found in cancer tumors, which abide by quite different mutational laws, and also there only upstream in the promoter region. And so the mutation data set that we used to identify this was obtained from sequencing hundreds of thousands of individuals of humans and basically comparing their genomes. And so this was one data set came from the UK, and one data set came from individuals that were sequenced around the world. And looking at these variants that differentiated individuals from one another, we can use a subset of them as a proxy for the germline mutational process. And this is what we use. So we call this population sequencing data as a part of an entire population of people is sequenced. So this is where we found this excessive hotspot. And then we tried to see how it looks in a completely different data set of mutations, which we assembled from family sequencing experiments. So in family sequencing, what is being done is the mother, the father, and their child or children are being sequenced, and then the genomes between everyone are compared. And any mutations that are present in the child, but not present in either mom or dad, are then designated to have arisen in this last generation. And the mutations that could have arisen there, these de novo mutations, include those that arose in the gametes of the parents. So that would be the oocyte in the case of the mom and the spermatocyte in the case of the dad. And also the set of mutations that arises after the formation of the zygote, right? Because they come together, they form the zygote. And from the zygote, the whole body of the child is built through cell division, right? So these two sets of mutations are different in one important way, namely that the mutations that were present in the gametes, they are present when the zygote is formed, and that means that they will be present in all of the cells of the body of the child. whereas the mutations that take place after the zygote formation, when these many cell divisions take place to form the adult body during embryogenesis and later, all those mutations will only be present in a subset of cells that later form the body of the child. And we call them, because of that, we call them mosaic mutations. And so what we did was we basically assembled from this family sequencing data sets, we assembled a data set that was only made up of these mosaic mutations, and we looked at their distribution around the TSS. This was motivated because we had another piece of evidence that I'm just not going to go into much detail now, but can be found in the paper, that told us that probably these mosaic mutations are enriched at the TSS. And this is indeed exactly what we found. We found that they are very, very strongly enriched immediately downstream of the TSS. So basically in one half of where we saw the hotspot before using this population sequencing data. No? So downstream into the gene body. So this led us to think that these mutations there downstream were predominantly caused during early development of the embryo. And yeah, so then we tried to find out what causes this hotspot, right? Both in upstream and downstream from a mechanistic point of view, because clearly it was a novel phenomenon, but we had no idea what could be driving it. And the way that we approached that was to basically calculate the association between 38 different epigenetic and genomic features and the mutational hotspot. So basically, how are these linked together, all these features with the mutational excess at the TSS. And one feature that was significantly associated with the hotspot was the occurrence of somatic double-strand breaks. So double-strand breaks are events in which both strands of the DNA double helix break at the same time. And that can be quite catastrophic for the cell if it's left unrepaired. And we actually have a whole array of machinery in our genome to take care of the repair of exactly this type of event. And what our findings suggest is that double-strand breaks that happen at the TSS are not properly repaired, and that this is part of what's driving this excess of mutations. Even in individuals in which DNA repair is functioning properly, right? So we're not talking, the data that we got were not coming from diseased individuals. So people who have normal functioning double-strand break repair, but at the TSS, something is preventing that from happening. This is what the data suggest. And then another feature that associated very strongly with the hotspot was RNA polymerase II stalling. So we talked about that RNA polymerase before and transcription, and I didn't go into so much detail, but basically during transcription, what happens is that the polymerase usually starts to transcribe a small bit of DNA at the beginning before it pauses. And during this pausing, it just sits there on the open DNA because the DNA double helix had to be opened up then for the to make space for the RNA polymerase that it can go along and read this one of the strands. So it sits there on the open DNA and waits for a signal to continue transcription into productive elongation. And we found that the longer this pausing, the more mutations accumulate. And there are some kind of intuitive reasons for that. The main one being that while the RNA polymerase sits here, one of the DNA strands, the one that's not transcribed, is basically hanging in mid-air or mid-cytosol, right? It's like dangling around. And in this single-stranded state, DNA is highly vulnerable to DNA damage. So this could be driving it. Another factor that we found to play a role in this context is the formation of so-called R loops, which are hybrids between DNA and RNA. So the freshly formed messenger RNA molecule that gets kind of spit out by the polymerase can hybridize back to the DNA, and again, preventing the closing of the DNA double helix back into its stable form, and thus exposing the two strands to mutations. So those are among the main factors that we found to be driving this hotspot.
So, which genes are most affected by these mutations?
Yeah, so that's the question that was on everyone's mind. The genes that experience the strongest mutational excess at the TSS that we found are actually genes associated with cancer, cellular respiration, so mitochondria, and developmental errors like abnormal neuron morphology, which is important for brain development, and abnormal limb development. So, yeah, broadly speaking, genes that are expressed and particularly used during early development, as well as the genes that might predispose to cancer. So that means that the TSS mutations can particularly predispose the person who happens to get one of these mutations in their genome to these diseases and or problems in their life. And looping back to cancer and what I said at the very beginning, so most of the cancer risk that we have in life comes actually from mutations that we acquire somatically, not through the germline, but by exposing ourselves to mutagens like tobacco smoke or UV irradiation. There are also internal mutagenic processes that just take place in all of our cells all the time that we can't help. And so all of these mutations that kind of arise during life, they are the main risk, the main driver of cancer. And so then tumor can arise when a cell is hit by the set of driver mutations that is required for the cell to start growing uncontrollably, and they need to be in very specific places in the genome. And now you can imagine if you need, let's say, 5 of these mutations to start a cancer, if you already receive one of them in your germline, because it's one of these TSS mutations that maybe happens to land in the promoter of a gene that drives cancer when this promoter is mutated, then it gives cancer like a bit of a head start, because then it makes it, you have this mutation in all of your cells, right? Because it's a germline variant. And that means that wherever this cancer may arise, the cell that ultimately becomes the cancer just needs to acquire one fewer mutations than it would have normally needed to acquire somatically. And so that's basically where the risk, where the two themes connect.
How do these hotspots impact health, and are these vulnerable areas eventually weeded out by natural selection?
Yes, so because of the types of genes that we just talked about that we saw are most heavily affected by the hotspot, there is a clear implication for health, right? Based on like cancer or developmental disorders. But more generally, also because of what I mentioned at the beginning, that protein coding genes are the most functionally important regions in our genome. And we're talking about mutations that happen in their regulatory regions or on them, right? So these TSS mutations are generally very likely to destroy something important. And then when we looked at their evolutionary fate, we actually found this corroborated. So we looked at how does this hotspot maintain over several generations in the population by looking at older genetic variants that still segregate in the human population. And we found that this hotspot is very quickly purged of its excessive number of mutations. So they are weeded out, basically, like you said. And just what that means is, on average, that the people who had such a TSS mutation were less likely to produce offspring than people who didn't. And this is how these mutations were then removed. But there's a very important subtlety here, because as you remember, we found that some of these mutations are mosaic, right? So they happen during development after zygote formation. And that means that selection against these mutations might not kick in directly, because the person who has such a mosaic variant, so when they are in the womb and they are an embryo, they might acquire such a mutation, but in a subset of cells of their body that cannot cause disease, cannot cause this disease that's driven by this mutation that the person acquired. So they don't notice it. But they might have also, in the subset of cells in which they have the mutation, might also include their gametes. So then they can pass it on to their children. And their children, because it's coming from the gamete, will have that mutation in all of their tissues. And if the mutation causes a disease in one tissue, in some tissue, it will cause the disease in the child. And even though the parent was healthy.
Well, I guess the big question is, how does this discovery change things for genetic studies?
Yeah, so there are many studies looking at the impact of mutations on disease, right? And that's often quantified by comparing the number of mutations or substitutions at a given genetic locus to the number that I would have expected to see if the mutations at that locus were completely inconsequential for disease. So this expected number it's what's called this neutral null model, right? So what would happen under neutral evolution? And then I can basically compare the observed number to the expected number. And if the observed number is much lower than my expectation, I would conclude that, well, this locus clearly seems to be important because evidently the mutations the mutations and substitutions that I should have seen there, the mutations that arose there, are not detectable anymore, which means that they were purged out of the population because they caused a fitness decline. They were deleterious to their carriers. So now for that comparison to work, you can imagine it's crucial to get the expected number very, very right. So my background expectation of how many mutations I expect. And the existing models are not aware of this hypermutation phenomenon at the TSS because it was just not known, right? And they don't factor this in, these crazily high mutation rates. So if we illustrate that with a simple example, imagine I look at a locus and I have a model that doesn't know about the TSS hotspot. And maybe my model then predicts that I should see on the promoter of this gene that I'm interested in, I should see 20 mutations under neutrality. And then I look at the promoter and I count how many do I see, and I actually see 30. Now, that would be, first of all, very surprising to me. But then my conclusion would need to be, because it's actually, I see an excess of mutations, would be that this promoter is not only not under constraint, so mutations are not being purged from it, but quite to the contrary, it seems that the promoter is evolving at unexpectedly high speed, which means it is actually adapting. It is undergoing adaptive evolution, which means that the mutations that arise there make the people fitter and more adapted to their environment. So that could, in theory, be, but we almost never see adaptive evolution taking place. It's very hard to detect signals of adaptive evolution. And the main reason for that is that most species are quite well adapted to their environment, such that a mutation is much, much, much more likely to destroy something that's working than to make something work better than it already does. So it's just like orders of magnitude different probabilities. So I would be very surprised to see a promoter evolving under such high positive selection, under such adaptation. So now if I have a proper model because I know about the hotspot, then maybe my proper model would actually predict that I should see in this promoter 50 mutations. And now I get the right effect size because 50 is my neutral baseline now, and I can compare that to the 30 that I observe, and I see, aha, the promoter is actually evolving under constraint, and mutations that arise there are being purged from these regions because they're deleterious. Exactly also what we found in the paper by studying older mutations. So this is how mutational models in the future can be built and should be built, looking at these regions with new eyes, right? Basically not saying, oh, these mutate like everything else in the genome. No, they mutate at an unexpectedly high rates, and lots of mutational processes that are going on there are, I just knew or haven't been described, and well, some of them have been described, but the drastic effect that it has is not present and taken care of in most models.
It's extremely important work that you and your team are doing, Donati, and I'm very grateful that you've been able to take the time out of your schedule to come onto the show today. So, what's next for you? What upcoming projects can you tell us about?
Yeah, so, well, thank you very much again for having me on the show. It was a lot of fun. And I think it's also just really gratifying. And also personally, I think it's really, really important that the public and scientists connect as much as possible so that there is not such a kind of gap of knowledge of what we're doing. So I was very delighted about all the reactions to the paper, because any communication with the public is good. Yeah, so we have several exciting ongoing projects. They are regarding cancer evolution, of course, but also species evolution. So the two topics I already mentioned at the beginning. And it includes developing novel methods for cancer driver locus identification, with a strong focus on moving away from protein coding genes. Because so protein coding genes that drive cancer, they are on a positive selection. They are relatively easy to detect. I worked in the past on a couple of algorithms that do this, but there are lots of other algorithms out there as well. What is much more difficult is to try and identify regions in which mutations can cause cancer that are outside of protein coding genes. So in the intergenic space or in intronic regions, right, which are the segments between exons. They are in genes, but you cannot use the same methodology. And so that's why it's much harder to tackle methodologically. And this is what we like to challenge ourselves. So that's what we are working on. We're also building new methodology to quantify constraint acting on protein coding genes across species, so at the level of the phylogeny. That would be more accurate than existing approaches. That would, for example, take into account mutational processes better than existing approaches. And we're doing that to then know about kind of which proteins are very important for fitness in general, but we're also having a strong focus on what does that maybe have to do with cancer predisposition. And then finally, we also keep being interested in mutational signatures. So those are the different mutational processes that can generate mutations in cancer, especially. And we recently built an algorithm that allows for the decomposition of a cancer profile, mutational profile, for example, into the underlying mutational processes. And we use that algorithm for further downstream analyses. And also, I hope that the algorithm will see the light of the journal soon, because it's been on the bioRxiv for some time, and we just need to kind of finish with the publication process. Yeah, so that's basically what's happening right now.
Donate WEGHORN, thank you very much indeed.
Well, thank you again. Like I said, it was a lot of fun.